Hi, my name is Massimo Pinzani. I'm professor of medicine at UCL and director for the UCL Institute for Liver and Digestive Health. One of the major problems of modern medicine is the shortage of organ for transplant, particularly heart and livers. In a joint effort between different laboratories at UCL, we want to address and try to resolve this important issue. For this reason, two eminent leading laboratories at UCL, cardiology and hepatology, have decided to join forces. The objective is to obtain human 3D scaffold from discarded human tissue, particularly heart and liver. We believe that the combination of the tridimensional structure with the composition of the complex extracellular matrix will allow the growth of stem cell in the scaffold, leading to a full organ regeneration. It's now a pleasure to introduce my colleague Daniel, who will give more technical detail about the project.
Hi, I'm Daniel Stuckey and I work at UCL's Centre for Advanced Biomedical Imaging, where I lead the preclinical cardiovascular research. So we're going to use novel therapeutic methods of engineering whole tissue constructs that we can graft initially into small animals and eventually into patients. Importantly, in this study, we're going to use novel imaging techniques such as MRI, ultrasound and PET to look deep within these 3D tissue constructs so that we can start to see molecular processes deep within the tissue as it's growing in the lab and also after we've transplanted it into the animals. Specifically, we'll use MRI and ultrasound to look deep within the tissue at structures as they develop in vitro. We're also able to test tissue perfusion and look at other processes deep within these constructs, both in vivo and while they're growing in the dish. We'll also use near-infrared fluorescence imaging and optical projection tomography, novel techniques which will allow us to look at molecular and cellular processes deep within the tissue. Additionally, we'll use nuclear imaging techniques like PET and SPECT to look at molecular processes deep within the tissue, specifically targeting processes such as angiogenesis, apoptosis, and the metabolism of the tissue. So this multimodality imaging approach will give us unprecedented insights into the molecular and cellular processes that are occurring in our tissue constructs as they develop in the lab and also after we've transplanted them into small animals.
Organ factory and bioengineering is the immediate future of medicine. Investing in imaging is key in this process. In this project, we will use imaging to guide our progress non-invasively. As a clinician, I'm really thrilled by these new opportunities. This can only happen at UCL.